And our last speaker for this session is Jim Trimmer from the University of California at Davies.
Thank you, and thanks to Andy and Kate for this fantastic conference. I'm going to be talking about ongoing efforts at this monoclonal antibody facility that I direct called NeuroMab, and this is within UC Davis. It started as part of my research lab and then branched off 10 or 11 years ago as a standalone monoclonal antibody facility focused on validating antibodies for neuroscience research. And I'd like to talk about just some general concepts as well as things that are special challenges, but also special opportunities for validating antibodies for use in the brain. So one of the things that I thought about before this meeting was just about what we talk about when we talk about antibody validation in more general terms. And I came up with this, and it's overly simple and maybe a little bit beneath the level of things that have been discussed here, but I think it's probably a reasonable synopsis of what we've come to think about with antibody validation is we really shouldn't be talking about, how was this antibody validated in terms of, well, it's validated by a single technique, as if that then transfers or translates into other techniques. But really, for what has this antibody been validated? And really, we need to be careful to just say, well, it's been validated for Western blotting, for immunocytochemistry, immunohistochemistry, et cetera. And really, even in a more limited sense, under the conditions that were used to prepare the sample, do the binding. And we've heard this over and over at this meeting, but I really think it's important to even correct our trainees, correct our colleagues, when they say, well, this antibody was validated by Western, therefore I can use it across different applications, to just make it clear that that's probably going to lead to or potentially lead to problems. And this is basically borne out by some of the things we think about when we validate our antibodies. We're validating them for people that are going to be using them in brain tissue, and therefore we need to validate them there. There's a lot of cell-specific post-translational modifications and then interacting proteins that are going to make it such that validating them against a naked recombinant protein or synthetic peptide might be an initial step, but that many antibodies are going to be binding to epitopes that are occluded within the cell. And then this carries through to how sample preparation conditions can affect this, whether it's through generating adducts that are going to interfere with antibody binding, or constrained locations where antibodies don't have access to their epitopes. So we really need to be careful how we talk about validating antibodies. Again, a theme that's been recurring in this conference, so that the end users are really certain of how the antibodies have been validated and under what conditions they either have or have not passed validation, and whether those are going to have any predictive value for samples that have been prepared under different conditions, or even binding that's been done under different conditions. And the concern we're always thinking about and that carries through with everything we do is, can we use a single application, one that is presumably easy and has a reasonably high throughput, to generate results that have any predictive value for things that are more difficult? And heaven forbid that we hit upon something that's anti-predictive. I mean, we could use that effectively, but if we know that, but heaven forbid it's inadvertently anti-predictive and we don't realize that, and we're putting things into one bin that are in fact highly enriched for the antibodies that we'd be most interested in. And we started back at NeuroMab, so I've been making monoclonals for my own research since 1981. And in the early 2000s, in the neuroscience community, there was a journal, the Journal of Comparative Neurology, that really started to highlight some issues with antibodies in use in neuroscience, especially in immunohistochemistry. And this was basically corresponded to the emergence of knockout animals and a spate of retracted papers in this journal where the antibody labeling that had been published did not change in the knockout mouse. And the editors started to kind of write to the readers and authors to try to highlight these, first talking about controls, and then really in 2005, instituting some rather draconian measures for publishing in their journal that are still in place today, and I'll talk about those at the end. We contributed an editorial on this, and then there's been quite a few others. And then a short time later was this kind of groundbreaking review by David Rim that talked about the process that they go through in their research lab before anybody's allowed to use an antibody within the lab, similar to what we heard about at AstraZeneca and other organizations now. And back at the time, back in the early 2000s, there were really very few commercial suppliers were discussing antibody validation. It would seem like everything's okay and there's not a problem, and why should we start talking about it if it doesn't exist? There was clearly a problem within the neuroscience community, and we were approached to because of our long track record in making monoclonal antibodies for our own research and with some of our collaborators, to set this up on a larger scale, where projects come through, initiated by the research community, suggestions, they go through NIH, and then we take on these projects, and then use kind of neuroscience-based screening to evaluate the antibodies from that. And then we were able to maintain low prices because the University of California, for the first time in its history, was able to come up with a royalty-free distribution scheme where they didn't charge any licensing fees or royalties to a contractor who also agreed to keep the prices low. And then because the R&D was primarily funded by NIH, we were able to keep the prices very low. And this has generated quite a bit of use from end users using these, and we're tracking fairly and accurately the use. It's about a paper a day that's being published citing our antibodies. And this is basically the procedure we use, similar to what we've seen for the antibody validation from the different groups that have spoken at this conference. Difference is that the projects come from community suggestions through NIH, and then the project gets approved, and then we take it on. We produce the immunogen, which is generally a recombinant protein or large fragment thereof, immunize mice, electrofusion, and then we perform two different ELISAs. One is against the immunogen on 96-well plates, and the other is transiently transfected cells that have been grown in 96-well plates. And then we take a rather large number of positives from those initial ELISAs, and we take them through these in parallel. So we do a confirmatory fluorescent ELISA using fluorescent subclass-specific secondary antibodies for IgG1, 2A, and 2B, so we can get some early insights into the subclass of the positive antibody in there. Then we do immunoblots on brain samples, immunohistochemistry on brain sections, and immunocytochemistry on transiently transfected heterologous cells. And again, this is on the entire positive pool, so we're not using any of these as a pre-filter for any of the subsequent assays. Then we do competition assays, so if the antibodies are of a different subclass, we can set up a matrix to see which ones compete with one another, so we can find antibodies that have distinct binding sites. We go into other samples. These are for the kind of selected few. Knockouts, human brain samples, knockout for immunoblots, immunohistochemistry, and then usually family members expressed in heterologous cells. The few selected ones are turned over under an MTA to our contractor to produce and distribute. We archive these at multiple sites around the US, and then we also are cloning and expressing these as recombinant antibodies. So here's just some of our data. So the upper left panel are scatter plots of the two different ELISAs that we use, where on the x-axis it's the protein ELISA and the y-axis is the cell ELISA. And then we try to find things that are in the upper right quadrant that represent antibodies that work well on the protein itself and also on the fixed formaldehyde fixed cells. But oftentimes we're taking antibodies that are in the upper left, the lower right quadrant, just to be sure we sample antibodies that have different characteristics. Again, we take from the 3,000 wells, which are polyclonal at this point, so there's probably 20,000 to 30,000 clones in these 3,000 wells that we assay by ELISA. We pick somewhere between 100 and 150-ish, screen them all by labeling rat brain sections, sagittal sections by immunohistochemistry. And then immunoblots on brain samples that we run a curtain gel and then make a blot that we cut into strips and then test each antibody on strips. And you can see here's a positive here, here's a positive here, and then here's a whole group of positives. So very successful project for antibodies that work on Westerns, where virtually every antibody we tested gave a positive signal to the right size. And then this is expression of the target protein in heterologous cells by transient transfection. And we like this because we have positive and negative cells in the same field of view. And we have a marker for the transfected cells. So it's simply correlation of the novel antibody to the marker antibody or GFP tagged protein as far as can the antibody distinguish a transfected cell from an untransfected cell. Again, a pretty low bar, but when combined with the native tissue analyses, provide us with some molecular verification of target specificity. We then go on to, for the selected subset, immunoblots on knockout tissue. Again, these are all brain samples. And then also species cross-reactivity. This is mouse brain, rat brain. And then human brain, you're not going to take a whole human brain and put it in a homogenizer, so we're working with regions. And this is where neuroanatomy and knowledge of target expression comes into play, because you're not going to expect to see a band from a region of human brain where that target's not expressed. So neuroanatomy comes into this very heavily, both when we're cutting brain sections, because if it's a protein that's only expressed in the cerebellum, and you're cutting coronal sections, you're going to cut a lot of sections that don't have the cerebellum in them, and you wouldn't want to use those to screen those antibodies, and you only want to use the sections that contain the region of the brain that expresses the target. And so this gets us into thinking about immunohistochemistry in the brain and some of the challenges and opportunities. Most of the proteins we work on have very restricted cell-specific expression in the brain. This is an example not from our work, but one that I just pulled out last night that I thought would be fairly familiar with most people. This is an enzyme called Tyrosine Hydroxylase, which is the key enzyme in the synthesis of dopamine. And there's only a few cells in the brain that make dopamine. And these are the cells that are impacted in Parkinson's disease. But in fact, the major region of the brain that's impacted is the striatum, because that's where the axons from these cells that are making the enzyme go out here and they make synaptic connections onto the striatum. So when you look at the protein, you see lots of protein in the striatum, whereas if you just look at the messenger RNA, which is restricted to the cell body, you only see it in substantia nigra, among other neurons here. So there can be a disconnect between the in situs and the protein labeling that can be complexing, but also can be informative if you know the biology of the protein that you're working on and what its subcellular localization can be. And at an extreme, if you have a protein that's expressed in a neuron in the motor cortex, right, the RNA could be here, and the protein could be way down here in the spinal cord. Or if it's in a spinal cord motor neuron, RNA could be in the spinal cord, protein could be down in your big toe. So it's a challenge, but again, if you know where to look, it can be very informative as far as reinforcing the specificity of the antibody labeling. So brain samples, neuroscientists are really generally want to use these in the brain. Relevant cell lines are just really not appropriate for the most part, and even primary cultures of neurons are very limited. People culture hippocampal neurons. They are usually very immature relative to what you find in adult hippocampus. And again, they're a very limited subset of all the cell types. You obviously couldn't use hippocampal neurons, this is a hippocampus here, to look at antibodies against tyrosine hydroxylase. That just wouldn't be present in those cells. So here's just some data that I'll go through very quickly. These are in situ hybridizations, and this is a lot of the in situ hybridization, maybe all of it that I'm going to show, is from the Allen Institute for Brain Science, where they've done an analysis of the entire mouse genome as far as patterns of gene expression in adult mouse brain, and then also some developmental stages. So these are in situs from their website. You can see very strong, this is hippocampus, very strong in situ signals in these cells here in a region called CA1, but very low levels in CA2, CA3. And then this is antibody labeling, and you can see that the regional characteristics of the expression hold, but the subcellular ones don't. So this is a protein that's shipped very efficiently out of the cell bodies where it's synthesized out into the dendrites of these cells. So we don't see any labeling that matches exactly where the in situ signal are or is, but is down in the dendrites and up in the dendrites of these cells. So it matches, but it doesn't overlap, but it matches from a cellular level. Here's another example. In situs, all in the cytosol of the cell body and the nucleus, and then a protein that's present only on the axon initial segment of these cells, so right where it emerges from the cell body. Again, cellular match, subcellular mismatch. Here's a simple one. This protein that's expressed only in these nuclei and the brain stem, bink, 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 protein, messenger RNA, everything's quite simple. And then here's just one example of a knockout. Another very simple case, we actually did the immunohistochemistry before we saw the in situ data, which hadn't been posted yet. We made this antibody that labeled the entire brain was completely blank except for this one patch in the hippocampus. And it was astounding, and we had many antibodies that fit this. So I'll say, when we screen 100 or 150 antibodies by immunohistochemistry on brain, we get a number of remarkable, amazing staining patterns. Only a subset of which actually corresponds to the target. So we're making antibodies against things that give amazing staining patterns, but aren't really target specific. This one, there was at least a couple dozen that gave this pattern, which made us think that this must be it. It was so unusual looking. And then when the in situs came out, this is a hemisphere of a brain here that you can't see because there's no labeling anywhere really except for the edge here, except this region here intensely, and then adjacent to this, and that's exactly where we see the antibody labeling. So this is a little harder one, and this is where having a PhD neuroanatomist on staff is crucial, because here we have an in situ signal that's present in the dentate gyrus. The dentate gyrus itself is completely blank, and you can see that adjacent to it, there's a large level of labeling. It turns out that this labeling is not at all from these cells, but it's actually from cells that are way over here somewhere in entorhinal cortex, whose axons are coming in here. This protein is in presynaptic terminals that are synapsing onto these cells. And this is actually the protein that's coming from these in situ signals. So it's a case of the protein being generated in the cells here, and then trafficked very efficiently out the axons to where it lives and functions. So if we saw labeling here, in fact, it would be wrong. And because this is an axonal protein, we know it functions in the presynaptic terminals. We have to know where the axons and terminals of these cells end up, which is over here, to know where to look for correct antibody labeling. So again, some challenges that I don't think we find in any other tissue that I'm aware of, where you have proteins that may be present centimeters away from the cells that are expressing the mRNA, and having to know the connectivity of the neurons and the pathways in order to be able to interpret the immunohistochemistry. One of the things I wanted to mention that's just specific for the approach we use, we still use mouse monoclonal antibodies. We're still attracted to this, and one of the reasons is the simple reality that from any given project, we generally get monoclonals of different IgG subclasses, which then can be mixed and matched with one another in multiplex labeling using subclass-specific secondaries. So very high quality subclass-specific secondaries. We use the ones from Molecular Probes, Invitrogen, Life Technologies, Thermo Fisher, I think in historical order of their existence. And you can see that you know, we can get very discrete, non-overlapping labeling, both in immunohistochemistry sections and on immunoblots by simultaneously applying 3 different mouse monoclonal antibodies of different IgG subclasses. And this gives us tremendous flexibility by bringing forward antibodies from each IgG subclass for each project, so our end users can really mix and match antibodies with greater flexibility than if they were rabbit antibodies, where you could never use two rabbit antibodies, even if they're monoclonals, in simultaneous multiplex labeling, because they don't have subclasses. A rabbit IgG is a rabbit IgG. So that's a very useful technique that comes out of this. And then I just wanted to talk a little bit about this idea of overlap between different assays and whether they're predictive for one another. So I'm going to show you this. We have not done a quantitative comprehensive analysis. I'm just going to show you some examples of some projects. So this is one recent project that looks pretty simple. So the large light blue circle is the immunofluorescence against the heterologous cells overexpressing the target. The internal circle here is the immunohistochemistry on brain sections. And again, we set out with guidelines for that target, what the expectations are going to be as far as which regions of the brain, which subcellular locations, and then which antibodies fit that criteria in our scoring system. So these are the ones that scored, I think, 3 or higher. And then in the hashed circle are the immunoblot positives. And you can see in this case, the immunofluorescence assay against transfected cells would have been quite predictive for antibodies that were going to work in these native cell assays. Immunoblots are an inclusive subset of the immunofluorescent positives, and the immunohistochemistry were as well. So if this was repeated project after project, we could use this immunofluorescence assay as a pre-screen. It's very easy. We do it on a 96-well plate for antibodies that are going to work in subsequent assays. This isn't always the case, and you can see here now, if we did that for this project, we would miss a lot of the immunoblot positives. Clearly, what we'd like for simplicity and power and lack of confusion from our end users is to just focus on the antibodies that work across platforms. So immunofluorescence, immunocytochemistry, immunoblot, immunohistochemistry. That's what we'd like to find each time. And we get various size subsets depending on the project. This is, again, just a subset of the projects we've done, and we haven't gone through these in any kind of quantitative way, but you can see that we generate projects that are all over the place. So that in this case, you know, we really couldn't use one assay to predict efficacy in a separate assay. So this leads me into finishing up. I just want to tell you quickly about a new project we're doing, and this is aimed at going beyond the complexities of neuroanatomy to looking at synapse complexity, which is hugely complex. And so in a mouse cortex, there's about 10 to the 9th synapses per cubic millimeter. So densely packed, really small. So there's about a synapse every cubic micron, and that's about the size of a synapse, just smaller than that. So they're packed in at incredible density, and they're each unique. And it's the molecular characteristics of the proteome that are within these synapses that underlies their function, and things like learning and memory and dysfunction in neurological and psychiatric disorders. So we'd really like to come up with a molecular definition of the proteomic composition of individual synapses. And a few years ago, Stephen Smith and his colleagues came up with a way to do this by using a technique called array tomography. And what this does is it takes a brain and it fixes it in plastic like you would use for electron microscopy, so low acryl or LR white. And then it cuts very thin sections. So these are 50 nanometer thick sections. They come out like a piece of film. So they come out as a ribbon. So serial sections that can then be stained, imaged, and then reconstructed into a 3D volume. And you could even put an EM step in here so that then you can paint the fluorescent signals that you're getting from the labeling onto this 3D reconstructed volume of ultrastructure level structure. The nice thing about this too is because the samples are embedded in plastic, you can strip and reprobe them many times over. So you can do multiplex labeling, image, strip, 5 more antibodies, image, strip, and generate this large proteomic, deep proteomic dimensionality of a large volume. And this is just some of the types of data that come out of here where you can look at individual synapses and look at the proteins that are present there and begin to classify these. And again, even at the EM level. The problem is, as you might have expected because of the somewhat exotic way these samples are prepared with alcohol dehydration and embedding in plastic, that very few antibodies work for this. So very few antibodies that are out there that are beautiful antibodies otherwise, because they've never been, people never thought about this when they were developing, characterizing these antibodies. There's no expectation that they would work for this technique, and in fact, most don't. So we've taken on trying to find those antibodies, and one of this reasons is because we took an old fusion that we had done, an old project way back in the mid-1990s, where the antibodies we were using didn't work for this technique. And we went back and rescreened because we archive everything. So we archive all 100 to 150 positives from the ELISAs in hybridomas in liquid nitrogen, samples in the freezer of the TC soups. And we've performed this retrospective screen 15 years later. And in fact, we found in our library that there were antibodies that worked for this technique that didn't exist back when we made the antibodies, but they were there. So we just embarked on a large project both to go back and look at our existing libraries where we have about 20,000 or 30,000 antibodies from all the projects we've done, as well as to generate new antibodies. And somewhat consistent with our previous findings for immunoblots, immunohistochemistry, immunocytochemistry, the antibodies that work for array tomography are not falling into any single class of these other types of assays. So none of these have much predictive value for antibodies that are ultimately going to work in array tomography. We just have to screen directly for array tomography. Now the problem is doing this on brain sections is excruciatingly painful, both dealing with these 50 nanometer thick sections, which are fairly wispy, but also the fact that it has to be done with 100x oil immersion, objective, visually, and trying to determine whether the classes of synapses that are labeling are in fact the class that should be labeling by comparing them to other markers. So we developed just recently an easier version of this where we just take the same heterologous cells that are transiently transfected with the target. We embed them we dehydrate them, embed them in the plastic, cut the same kind of ultra-thin sections, attach them to 96-well plates, and then we can process them for immunocytochemistry. And it gives us a much more kind of thumbs up, thumbs down. Yeah, this antibody works under these conditions. And this ends up having an almost, 100% predictive value for antibodies that are then subsequently going to work for array tomography. So I think I'll finish up. I just want to point out that, again, the Journal of Comparative Neurology has been on top of this. I'd like to commend them for over a decade, this idea of antibody validation. They still have these very high standards. I talked to the editor about what the feedback was from authors when they first instituted these standards for what it took to publish a paper there. And he said, well, there were quite a few people that were mad, and they said that they would never publish in our journal again. And in fact, he said we were happy to see them go, because if they weren't going to adhere, if they couldn't or wouldn't adhere to these standards, then they shouldn't be publishing in that journal because there was a higher likelihood that what they were going to be publishing was incorrect. This is the kind of information you had, so you asked to provide a table. We're now requiring our ID numbers from the antibody registry, catalog information, epitope or immunogen information, the amount that was used in the assay, and then you're also asked to write a paragraph on each antibody so that you can describe how it was validated. And then just last week, Cell Press came out with something very similar. So I don't know if you've seen this, but these new STAR methods that have replaced experimental procedures for structured, transparent, and accessible recording. So some of the same ideas here: catalog number, RID number, source, antibody. So this is a very, very dramatic shift for Cell Press from pushing people to eliminate as many methods as possible for space constraints to kind of opening things up, and I think this is a really good step. So focus on antibodies for specific applications, describing these conditions transparently. And as I think I mentioned yesterday in the discussion, I think this kind of makes an implied contract with our end users that if we describe exactly how we validated them and the conditions we use, they should be able to reproduce the results on our data sheets. If they change those, then our responsibility's over. And we have some special considerations that have come to the front in these studies of synapses. I'd like to acknowledge Randy Stewart at NIH, who's been the program official there that's been monitoring us and overseeing us all these years and been very supportive. Folks in the NeuroMab facility in my lab, and then people involved in this Synaptome project. And thanks for your attention, and happy to answer any questions.
Fascinating talk. Thank you very much. We're a little bit over time, so perhaps maybe just one or two questions? Or, yes?
I was just wondering, because going back to the Journal of Comparative Neurology, and it's taken a long time for others to follow on, and I'm just wondering if we've got any comment on why that is, and is it a reflection on how successful it's been?
That's a really the Journal of Comparative Neurology is a very high caliber journal for what it is, which is comparative neuroanatomy. So it's not, it's not part of the popular press. So I think it was viewed as, an extreme standpoint by a bunch of hard-ass people that were just they could do that because that was their little niche, but it wasn't really applicable, to your general run-of-the-mill cell molecular biologist. It was a neuroanatomical, I think that's probably the case. Yes?
Quick question. So you mentioned that you still like to use mouse monos so you can separate on the IgG class, and I can completely see the advantages of that. Just to say, in our hands, we generally find our rabbit monoclonals are much more sensitive and specific. I'm just wondering whether you completely exclude looking at rab monos, whether you could be
We've actually made some from some of the rabbits where we had beautiful polyclonal serum that we were using as affinity purified antibodies. We made some recombinants, but for us it was just the amount of cost and effort relative to the limited utility for multiplex labeling, and then the thought of re-engineering those with different constant regions to try to acquire different detection flexibility and stuff. It was like, well, we're making these and we're getting these anyway. When we make our recombinants, we are doing some additional swapping so that we can provide each of our antibodies in all three flavors, as it were, to increase flexibility. But and yes, so it's not to say we won't go back to rabbits. We're actually going the other direction. We're going into nanobodies and DARPins and monobodies and things like this. So thanks.
So Jim, you showed examples where you can predict the staining pattern based on knowledge from another assay, which in this case is mRNA expression somehow. is this, can you always predict, can you always have unique staining patterns that you can predict that will be specific enough so that you know that your antibody staining is specific?
I mean, yeah, so there are clearly, obviously, proteins that are expressed in every neuron. So histones, right? I mean, so there's going to be proteins that fall into that category where we're not going to be able to use that as a, I mean, they're the only criteria we would have is it's expressed in every neuron, and it's expressed in the nucleus or something. Most of our projects have not fallen into that category, so we can use this to our advantage. Again, sometimes it's easier if it's expressed in every neuron in the brain. It makes screening, certain aspects of the screening much easier, but that's not so typical except for real fundamental proteins that you would find probably in cells throughout the body, which is not most of what we're working on.
Okay. All right. So thank you to Karim and Chris and Jim for great talks, and it's time for coffee.